All right, hi guys. So this video is continuing on with our cell unit. Last week we looked at cell structure, and today we're going to start looking at the cell function. And one of the most important pieces of cell function is just how cells get materials in and out of the cell itself, or the cell membrane. So we call this cellular transport. And so the organelle that plays the biggest role, of course, is the cell membrane. So as a reminder, the cell membrane is what regulates what enters and exits the cell. This is where a cell takes in food and removes waste, and it also provides some support and protection to the cell. Just a reminder that the cell membrane is present in both plant and animal cells. So all kinds of cells have a cell membrane. So if you look here, the picture on the left is representing a simple animal cell. And so the cell membrane is this round, flexible barrier. We learned in plant cells, however, plant cells do have a cell membrane, but they also on the outside, they have a thick layer called the cell wall made of cellulose. So, and remember that the cell wall is only found in plant cells. Looking a little bit deeper into the cell membrane, we actually call it a lipid bilayer. So you know what lipids are, and then bi means two. So this is a double layer of lipids. The lipid bilayer gives the membrane a tough, flexible structure that forms a strong barrier between the cell and the surroundings. Within this lipid bilayer are three types of molecules kind of plopped throughout, and you'll see lots of different diagrams, even just in this lecture today, of what the lipid bilayer looks like. So plopped within this cell membrane are the lipids, and the lipids provide the waterproof protection. There are proteins, and the proteins help materials cross through the membrane. And then there are carbohydrates, and the carbohydrates here allow cells to identify each other. So I kind of think of the proteins as the gates, and you'll see this in just a second, but I think of the proteins as the gates through the membrane, and then the carbohydrates are kind of like the gatekeepers. Looking at this diagram down here, the lipids are these structures that kind of look like a sperm. They're typically thought of having actually two tails each. So here's one lipid, and here's one layer of the lipids, and then a second layer of the lipids. And so that's why we call it the bilayer. So there's two layers of the lipids there. And then plopped among these lipids are proteins. So this right here, this like tan looking structure is a protein. And you can see it is actually structured to allow materials through. So it'll come, if it's allowed to pass through, a molecule may come and pass right through the protein. And then there's also carbohydrates, we said, and carbohydrates are shaped in lots of different ways. And so I always just remember that the carbohydrates look kind of goofy. So this is an example of a carbohydrate here, but then also plopped in the lipid bilayer are two carbohydrates here. And these again act kind of like the gatekeepers. So they really just are constantly monitoring the surroundings and identifying, like as a cell kind of moves around, identifying the surroundings. Here's another diagram of the phospholipid bilayer. So remember, in this instance, the lipids themselves are these orange and yellowy shaped figures. There's a top layer here and then a bottom layer here. So that's the bilayer. And there's a lot going on here, but these big gold blue blobs are the proteins. And actually the inside of those proteins are going to look like this right here. So you can see that it is literally structured to allow a substance through. So either going into the cell or out. And then the funny looking things like these green molecules or the yellow molecules or these funny looking blue molecules, those are the carbohydrates. And the carbohydrates, remember, are basically just constantly monitoring the surroundings, and anything that comes into contact with the cell, they're trying to identify what that is. Keep in mind that every living cell is filled with liquid and is surrounded by a liquid. So even organisms, plants or animals that are in the driest, hottest of deserts must maintain liquid within the cell itself and surrounding it. So we're going to talk a lot about the concentration of a solution. And I've kind of tried to simplify it here at the start. So the concentration of a solution is the amount of solute in a given amount of solvent. So the solute is what is going to be dissolved in the solvent. And typically, we think most of the time we think of solvent as being water. So I actually like the image here on the bottom right best that shows two different sized beakers. Let's imagine that the blue solution is just water. And you know what, guys? We're going to pretend that those yellow dots are sugar. Let's say we're making Kool-Aid. Oh my God, I haven't made Kool-Aid in, a really long time, 15 years. So let's imagine that we have a smaller beaker here and then a larger beaker, and we've put some water in there, and then we've added some sugar. Let's say we're making Kool-Aid. If you were to sit here and count, there's actually the same amount of yellow dots in each beaker. So again, let's imagine it's the same quantity of sugar, okay? If I were to stir those beakers up and give a taste test to my twins and say which one tastes better, they would probably like the smaller container because it has the same amount of sugar and less water. So it would taste much sweeter. So in this instance, the sugar would be much more highly concentrated, right? So there'd be a higher concentration of sugar. If you look in the bigger beaker, again, it has the same amount of sugar, but way more water. My twins would probably not like that as much because it doesn't taste as sweet. So that is an example of the concentration of a solution. A classic example, might make you feel weird, is urine. So a lot of us probably don't spend too much time thinking about the color of our urine every day, but it is one sure thing that we do every day. So we all pee every day, and it is one way that we can kind of monitor our hydration. So a lot of us don't pay too much attention. A lot of us probably drink a healthy amount of water and we pee and move on with our day. But it is a really common, what am I trying to say? It is one piece of a lot of problems. You monitor the color of your pee. This might sound weird, but a lot of older people, once people get like older towards the end of their life, they have no desire to drink a lot of water. It does not sound good to them. And they battle frequent like urinary tract infections because they're not drinking enough water to flush out the waste. And so bacteria builds up. And so literally just monitoring the color of their pee is one of the the easiest ways to monitor hydration. So as we start to look at membranes, we will see that some cells have what we call a permeable membrane, and then some are what we call impermeable. And so permeable means that substances are able to diffuse across the membrane. Impermeable means that the substances are not able to diffuse across the membrane. So the prefix im means non, so that's not permeable. So if you look here at the videos at the bottom here, the person on the left is representing an impermeable membrane to water. So that person has a poncho on, and they're intending for that impermeable membrane to keep the water, the rain out. A permeable membrane would be like regular clothing, and which very much would allow water through. So we have permeable and impermeable membrane. Then we have selectively permeable, and most cell membranes are actually what we call selectively permeable, which means that they're going to allow some substances to pass through and others not. So they kind of pick and choose. And really, you'll hear me say this over and over, but really, there's a pretty simple rule. So if you look at the image here, the lipid bilayer is represented by the blue and red molecules. So here we are with the cell membrane. And the molecules down here at the bottom are examples of materials that are usually not allowed through. So without the help of like a special protein or something, which we'll get to, usually these are not allowed to just freely pass through the cell membrane. If we start at the very bottom, we're looking at ions. And remember, ions are any molecule with a charge, a positive or negative charge. And because they have a charge, they're not going to be allowed through. Now, you'll see in a bit that there are ways that we will, like special protein channels, for example, that will pump these things through the cell membrane, but it's trickier. The other tricky thing, you guys, are large molecules like glucose and sucrose. These big, bulky molecules are typically not allowed to just freely pass through the cell membrane. They need some help. The two groups here on top that you can see here are much more freely allowed to pass through the membrane are smaller molecules. Okay, so smaller molecules without a charge. Examples of this are like oxygen, carbon dioxide, nitrogen, even water. So smaller molecules that don't have a charge. We will look at 3 types of transport. So 3 different ways that cells can move materials across the membrane. We'll look at passive transport, facilitated diffusion, and active transport. So I love this diagram because it kind of ties it all together before we take a closer look. So with passive transport, there's 2 examples of passive transport. And with passive transport, you guys, I want you to think truly of like a passive person, a very laid back person. So passive transport doesn't require energy. Okay, the cell, the molecules can just move through the membrane pretty freely. Active transport over here on the far right is just that. It is active, and think of it as being active. So active transport requires energy for these molecules to move through the membrane. So let's take a look here. With passive transport, this is the movement of a substance through a membrane without the use of energy. So passive transport does not require an input of energy. Examples of passive transport are diffusion and osmosis, and we'll look at those in just a second. We'll look specifically at diffusion and osmosis. The key here, you guys, the key to passive transport is for molecules to pass through the membrane with no energy is that they have to follow what we call the concentration gradient. Molecules naturally want to go from a high concentration where they're squished together to low concentration. So if you look at this image, the lipid bilayer is right here down the middle, and the molecules that we're looking at, you can see that there's a much higher concentration of them here on the left side. So there's a lot more of these funny looking molecules on the left side. And so naturally, these molecules are going to want to move from high concentration on this side of the membrane. They're going to want to try to pass through the membrane and go to the lower concentration where there's only 3 of them. And so you guys, this is what's going to happen. Literally, the molecules are going to go from high concentration over here and pass through the membrane over here. And it's going to keep moving in that direction, these molecules, until you guys, it's the same. Essentially until there's the same on the left side as there is on the right side. And we'll get to that again in just a moment. So here you can see the molecules, it actually wasn't even working last time. Can I get it to go again? No, that's okay. The second type of passive transport is what we call facilitated diffusion. And so with the term facilitated, I think of as like a facilitator or like a teacher or a helper. So with facilitated diffusion, this is movement of specific molecules across cell membranes through protein channels. So this is when a molecule cannot just freely pass through the membrane, but they need a protein channel. And this happens all the time. There are protein channels everywhere in the cell membrane. A lot of stuff moves through facilitated diffusion. So if you look at the image here, the phospholipid bilayer is like the little blue looking molecules right here. And then plopped down among there are these big old purple protein channels. So this is representing facilitated diffusion of the red squares, you guys. So if we start here on the left, this protein channel, so it's going to be tightly closed up, and it's definitely monitoring what is trying to pass through. And they're really specific, kind of like an enzyme, whereas these guys only allow a particular molecule through. So this purple protein allows red squares to pass through. So the purple proteins are hanging out. Red square comes, it gets the thumbs up, and essentially the purple protein passes the molecule through the membrane. So you can see in the image here, the red square begins its journey through the protein channel. It continues through until the red square makes it to the inside of the cell, and that's a good thing. So these red squares, this is not requiring energy. Notice, you guys, that the red squares are moving from an area of high concentration to low. So these red squares, there's a lot of them outside of the cell, and so they want to move into the cell. And because they're following the gradient, it does not require energy. So these protein channels allow molecules that cannot pass through the membrane to diffuse into the cell. And again, it does not require energy. Here you go. You can see these orange squares are moving from an area of high concentration to low. So they're moving through the purpley colored protein channel from an area of high to low concentration. A real life example of this is glucose. So glucose is a pretty big and bulky molecule, and glucose is, we'll learn actually in our next unit or next section of notes, how important glucose is for our cells. But so glucose must enter cells constantly, like all the time. Our cells need a huge amount of glucose, and but it cannot freely pass through the membrane because it's a big bulky molecule. But there's a really special protein that's plopped in the membrane, and it allows just glucose. So its only job all day, every day, 24/7 is to just find a glucose molecule and pass it through. So this protein is plopped there, just grabbing a molecule of glucose and passing it through, right? So just pumping glucose through the membrane, for example. And there's hundreds of examples of this in our body. So different protein channels are going to allow specific ions, specific sugars, salts, whatever it may be, in or out of our cell membranes. And then the third type is active transport. And again, think of active transport as truly being like an active process. So here's the kicker here. The movement of the materials across the cell membrane are going against the concentration gradient. This is the first time that we have seen that the molecules are going against the gradient, which means the molecules are going from an area of low concentration and pushing it to high. Because it's going against the gradient, it requires an input of energy. Think of it as needing like a boost to move through. So in the diagram here, you can see this is the lipid bilayer, and plopped here is a protein, a purpley protein. And we call this specific type of protein a proton pump. So hydrogen protons are going to be moved from one side of the cell to the other. And in this instance, you guys, notice it's going against the gradient. So for the first time, we have just one little hydrogen here, and it's actually being pumped to an area of much higher concentration, of hydrogens. And because of that, it requires a boost or like an input of energy. So it's going to require the cell to use energy. So this goes against diffusion. It requires energy and it pumps materials in and out of the cell. The input of energy, it's kind of important to note, I think, if you look here at the picture, the yellowy like star molecule is labeled ATP. That's what we call cellular energy, and we'll learn about that in a couple of days. ATP stands for adenosine triphosphate, and it's just the like literally the energy that our cells use. And there's a special process in our cells that make that energy. So they basically take glucose energy, like from the lunch I just ate, and our cells will take energy from my lunch and turn it into ATP. And so the ATP is really what fuels all of these cellular processes. So active transport requires just an input of ATP. Okay, so you've heard the term diffusion, and again, this is a type of passive transport. Does not require energy. Just this is the way that molecules most often want to move, is from an area of high concentration to low. An example of this, so in the definition of diffusion, you can see that this is the process by which molecules move through a membrane from high concentration to low. So most often, diffusion is talking about molecules moving through a membrane, right? Like moving through the cell from high concentration to low. Sometimes the examples are like, even like cologne, right? So, I mean, every year there's one period a day where a high school boy comes into my classroom every day, the same period, just reeking of cologne, right? And so this person has sprayed cologne all over him. And the reason that my whole room starts to smell like this kid is because of diffusion. So those molecules essentially are stuck to his clothing, and they naturally want to go from an area of high concentration on his clothes to low. And that's so it's diffused, the cologne is diffusing through my classroom. Although in that example, it's not passing through an actual cell membrane, that's essentially what molecules want to do, is go from high concentration to low. I don't think this works. No. So diffusion causes substances to move across the membrane without the use of energy. Remember, this is passive. Molecules always go from high to low concentration. And again, when the concentration is the same, we call that equilibrium. So I kind of hinted on this earlier, like what happens when we're making the Kool-Aid and the concentration is the same on either side of the membrane, right? We call it equilibrium. So when concentration becomes the same on either side of the membrane, that is equilibrium. So let's look at an example here. We have two different substances on either side of the lipid bilayer. So here, like these goldy looking structures is the lipid bilayer. And then we're looking at two different molecules. So we have green circles, or I'm actually going to call these little blue ones water molecules. They look like water molecules to me. So within this diagram, we have a cell membrane right here. And we have two molecules that are going to try to go in one direction or the other. So what's going to happen, you guys, is if I were to sit here and make you count the green circles, you could see that on the left side of the membrane, there's 4 green circles. And on the right side of the membrane, there's way more green circles. So because of that, I know that there's a higher concentration of green circles here on the right side of the membrane. So the green circles are going to try their darndest to go from the right side of the membrane and move to the left. So they're going to go from the higher concentration of green circles and move to the lower. Again, eventually, it'll get to the point, you guys, listen up. It'll get to the point where there's the same number of green circles on both sides of the membrane. And that's what we call equilibrium. But In reality, it doesn't just stop. The molecules don't just stop on that side. Truly what happens is once the same number of green circles are on either side, one green circle may move this direction, and then another will move in this direction, and one will move in this direction, and one will move in this direction. And it maintains that equilibrium. So stuff is still going to continue to pass through the membrane, but it does it at an equal rate, always trying to maintain equilibrium. Let's take a sec here because I think it's kind of important that if we look at the water molecules or the little blue molecules here, you can see that they're different than the green circles. So we look at the little blue water molecules. There is way more water on the left side of the membrane than over here. So in this case, we're going to see a lot of water molecules moving from high concentration here and moving out of the cell. Again, water molecules are going to move out of the cell to the right until there's the same number of water molecules on both sides of the membrane, and then it will try to maintain that equilibrium from there on. The term osmosis refers specifically to the diffusion of water. So osmosis is always water. So the diffusion, so water is still going to be moving through a semi-permeable membrane here, but it's only water. So nothing else is going to be moving through except water, if we're looking specifically at osmosis. I like to show this picture because it shows how desperately molecules want to reach equilibrium. And so there's this special, I forget what they're called, I forget it every year, but there's this special glass tube, a piece of scientific equipment that looks like a U. And at the very bottom, scientists can actually place a semi-permeable membrane. So imagine that this black dotted line at the very bottom of the U there is a membrane. And in this instance, it's a semi or selectively permeable membrane that only allows water through. And the scientists know this. So when they're setting up this experiment, they know the type of membrane they've plopped there is only going to allow water. So what they've done, you guys, they've started this experiment on the left side. And they have dropped solute. So the purple circles are the solute. So you can see that the left side, you guys, has less solute than the right side, right? So this is much more concentrated with the purple dots. Again, if you think of it as sugar and Kool-Aid, this would taste much more sweet, right? So it would be much more concentrated. If in this special piece of equipment, if we give it time to reach equilibrium, what the water wants to actually happen is maintain the same concentration on either side of the membrane, right? So we want, these molecules are going to try to move, desperately try to move, to try to reach the same concentration on both sides. So if you look here, you can see that the roughly, I won't make you count them, but roughly, if you picture it as sugar and Kool-Aid, you guys, the left side here is going to be just about equally as sweet as the right side. And it's because the right side took on more water molecules. So what we see happen, you guys, is actually the water molecules want to reach equilibrium so bad that they actually defy gravity, and they actually can move through the membrane until equilibrium is reached, until the same concentration is on the left side of the membrane as the right side. Pretty amazing. So when we're looking at the solutions and the concentrations, we have names for them, of course. So a hypotonic solution is a solution in which the concentration of solute is lower than inside the cell. With a hypotonic solution, the water is going to rush inside the cell. A hypertonic solution is when a solution is a solution in which the concentration of solute is higher than inside the cell. And with a hypertonic solution, the water is going to rush outside of the cell. And we'll look at this lots of times. And then an isotonic solution is a solution in which the concentration of the solutes is the same inside and outside of the cell. So with an isotonic solution, water really isn't going to move. So let's take a look at these. So this is osmosis because we're looking at the movement of water. And so what happens with a hypertonic solution, water is going to leave the cell. So the water molecules are going to go from an area of high concentration to low. In this instance, the water is actually going to leave the cell. I think of hypertonic as I think of the water molecules as feeling really hyper, like they got to leave the cell, and that's what they do. And we can actually see cells become like shriveled and weird because they lose enough water. Isotonic solution, we said, is the same, so it's at equilibrium. If you look at the diagram here, we oftentimes show an isotonic solution with an arrow going both in and out of the cell, you guys. And that's because I said a lot of times with equilibrium, there's still water moving in, but then the same amount's going to move out. So it's like this constant maintaining equilibrium. So with isotonic, the size isn't going to change. Water's coming in just about as fast as it's going out, right? Just constantly maintaining. And then with hypotonic solution, water is going to rush into the cell. So because of the solution the cell is plopped in, water is going to move from a high concentration outside of the cell to an area of lower concentration inside. And so we actually see cells getting bigger and bigger and bigger and bigger because they take in water. The term that creates this kind of like the pressure of this movement is called osmotic pressure. So this is the pressure on the hypertonic side of a selectively permeable membrane. So this osmotic pressure, you guys, is what causes a cell to either shrivel or expand. And we can see cells shrivel so much that it dies, or cells actually get so huge and big that they burst, right? So osmotic pressure can cause the cell to actually die. And there's a term for that, of course. So plasmolysis is the shrinking of a cell from losing too much water. So plasmolysis is a cell shrinking so much it can die. And then cytolysis is the bursting of a cell from taking in too much water. So cytolysis is the actual bursting of the cell. This little table just kind of sums it up, reviews what we've already talked about. So let's do a few practice examples. So if you guys look at these pictures, this is a beaker with a solution and then a cell plopped in that beaker. Here's a big old cell. And I have given you the concentration or the ratio of water and solute in both the cell and the beaker. So let's look at the first one here. The solution is 85% water and 15% solute. And then the cell has 90% water and 10% solute. And what we're looking at here is the water. So I asked you, which direction would the water move? That's key. So we're looking for the higher concentration. In this first example, there's a higher concentration of water at 90% in the cell than 85% out of the cell. So the water is going to move from the area of higher concentration in the cell out. So if I could draw, I would draw an arrow from in the cell moving out. If we look here at the middle beaker, we're looking at the concentration of water, and there's 90% in the solution here and only 40% water in the cell. So water is going to go from the higher concentration, which is 90%, and move into the cell. The third one here, there's a 80% water in the beaker and 75% in the cell. So the water is going to move from higher concentration into the lower concentration, which is in the cell. So here are my arrows. Remember, we said the first one, the water is going to actually move out. And we call that hypertonic. Remember, I think water molecules is feeling really hyper, like they want to get out of the cell. And then hypotonic, water is going to move into the cell. Let's do a couple more examples. So we're, again, still looking at the water movement here, and the percent of concentration in both locations. So the beaker here is 75%, and in the cell is only 45. So it's going to go from the higher concentration and move into the cell. The middle one here, there's a higher concentration in the cell because it's 90% compared to 63 in the beaker. So water is going to go from the higher concentration and move out of the cell. Whoops. Oh, darn it. And then notice the last one here is actually the same. So because the concentrations are all the same everywhere, we're going to call that isotonic. And notice my double arrow. So water is going to move, remember, move in at the same rate that it's going to move out and just maintain. Here are some real microscopic images of cells. I think this is so cool. So remember, we have in the middle here's isotonic, where it's equal. So these are like healthy, normal cells. Hypertonic cells lose water and shrivel. So here you can see actual cells shriveled. And then you can kind of tell that over here on the far left, those are solutions in a cells in a hypotonic solution. You can kind of tell they're a little bit bigger maybe, but it's not as easy to see there as the shriveled. Okay, hold on. Sorry, I'm gonna pause and take care of my baby real quick. Okay. There we go. Okay, so there are two ways that cells try to prevent osmosis problems. One of the ways that cells can do it is by having a cell wall. So remember that plant cells have cell walls, which make a rigid, more protective layer. And that cell wall will prevent them from bursting. And then some cells have contractile vacuoles that are just constantly pumping or maintaining the water in the cells. There's also bulk transport. So instead of moving like individual molecules through the membrane, like we've been focusing on, there is bulk transport, and this is just the movement of large amounts of material through the membrane. So we have endocytosis, which is taking in a large amount of material. And then there's exocytosis, which is removing a large amount of material. And it is actually pretty simple. So it really is as simple as like this diagram almost. So remember, endocytosis is bringing in, and truly the cell membrane you can see there. So the cell is like this sky blue looking blob. It truly does just kind of like use its flexible cell membrane to kind of engulf whatever it wants to take in, and it just kind of brings in the material. So let's look at, I like this image because it shows both. So you can see this is the cell membrane, and it truly does just kind of like make a little pocket in the cell membrane. Call that a vacuole for like storage. And then it takes in the molecules that it wants, encloses it, and then moves it into the cell. And it does the opposite to get rid of it. So exocytosis, it's going to collect waste in a little storage vacuole, and then eventually literally just move. You can see here this pocket of waste. It's going to move it to the cell membrane and essentially just kind of like spit it out, get rid of it. So to review, there's three types of transport here. We have diffusion right through the membrane. Facilitated diffusion, which is using a big old protein channel to allow molecules through. Both of these types are passive transport because it's moving. Look at both of these molecules here are moving from an area of high concentration to low, right? So that does not require energy. Active transport here on the right does require an input of energy in the form of ATP. And notice that the molecules are going from an area of low concentration to high, which is odd, and is going to require that energy. So I've talked a lot about maintaining equilibrium, and we have a term for that. So the movement of all these things are constantly being maintained, and we call that homeostasis. So homeostasis is the ability or the tendency of an organism or cell to maintain like internal equilibrium by just constantly adjusting its processes. So that's what cells are going to do, is just constantly try to maintain this equilibrium. Okay, we're going to stop there with the notes and review a little bit together. So you should be done now with the second section of notes. I'm so sorry for the baby in the background. As you may recall, my husband had surgery and he literally can't pick up our baby for 2 weeks. So I can't go to school. I can't really do anything without the baby being there. So thanks for your patience. Email me if you guys need anything. All right, see ya.